Hey everyone. In this video, I'm going to talk to you guys about sexual differentiation during fetal development. Now, before I get into the process itself, I want to clarify just a little bit um, about what I mean by sexual differentiation. I want to emphasize that what I will be talking about is anatomical sex. So essentially, what is the genetic, so XX versus XY, or even XXY, or something else. Um, and in turn, how the physical structures develop in relation to those genetic genotypes. So things like the genitals, the hormones that are produced, different secondary sex characteristics such as body hair in different places, breasts of different sizes, etc. So we would call that generally the anatomical sex, but as we all know, there are many different types of, or many different ways of expressing something to do with sexuality. And so before I even start, I just want to emphasize that we are talking about anatomical sex. I am referring to male and female genotypes and phenotypes, but not gender. And so gender identity is um, your sense of gender. So you can identify as male or female or non-binary or anything else. So think about your pronouns. So that is your gender identity. He, she, they. Gender expression, on the other hand, is essentially how you want to express yourself, the clothes you wear, the activities you participate in, and even that is different than what you're attracted to, so your sexual orientation. And so if you have questions about this, I encourage you to refer to the genderbread.org site, which is what I have borrowed all of these images from, just to clarify that there are different types of sexual identity. Again, I am talking about anatomical sex, and this is separate than gender identity. And so ultimately, the male and female phenotypes, right? So the differences in genitalia, the differences in germ cells, so sperm versus egg, that is all influenced in many ways by what hormones we express and what hormones we produce the most of. And so I just want to clarify that all of the sex steroids are derived from cholesterol, and these aren't the only ones that are derived from cholesterol. Remember from other lessons that we also have cortisol, right? So our stress hormones are also derived from cholesterol, as well as things like aldosterone. And aldosterone is another steroid hormone derived from cholesterol. And so we have a series of enzymes coded for in our DNA that will ultimately determine whether we produce cortisol, aldosterone, testosterone, or estrogen, or any other of the steroid hormones. In general, whether you are genetically male or genetically female, you are always expressing, right, you're always producing both testosterone and estrogen from cholesterol. And there are a series of other sex steroids as well, and I'll talk about one of those today and then more in future lessons. But in general, we're all producing all of these types of hormones. Okay, so it's never that we synthesize all estrogen or all testosterone. Side note also, I will be referring to testosterone as T and estrogen as E2 or estradiol, but there are other hormones as well. Okay, so not only do we produce both testosterone and estrogen and the others, but we also use both of these hormones to guide different processes throughout our body. For example, in males, estrogen is used to promote sperm production. No estrogen, no sperm. In our bones, male and female, there are only estrogen receptors, no receptors for testosterone. And so in order for testosterone to promote bone growth, it actually has to be converted into estrogen before it can actually bind to the bones. Okay. And so that leads me to talk about enzymes. In order to go from cholesterol all the way down to estrogen, we need a series of enzymes. And ultimately, upregulating each of these different enzymes is going to produce more or less of these different hormones. So, for example, androstenedione is a common precursor androgen, or what would be referred to as a masculinizing hormone, but it can be converted into testosterone, right? So some basic changes here, pretty much the same, except for androstenedione has just an oxygen here and testosterone has an extra hydrogen. Okay, so the enzyme responsible for catalyzing that reaction from androstenedione into testosterone is 17 beta HSD. Okay, to go from testosterone to estrogen, right? Again, very minor differences here. So we're missing one carbon, right, and a hydrogen over here, an extra hydrogen over here compared to testosterone. So super similar between these two hormones, which we would otherwise consider to be, very, very different. And there's a single enzyme that's responsible for converting T into E2, and that is called aromatase. Sometimes it's called CYP19. That's more referring to the gene that codes for this enzyme. Okay, so as you're researching, as you're studying, you might hear aromatase or CYP19. Essentially, we can think about them as being the same. Okay, so if we want more estrogen, we would produce more aromatase. Okay, if we want less estrogen, we would produce less aromatase. Okay, so let us start our story of sexual differentiation in week 5 of fetal development. That is about 5 weeks after sperm meets egg. About a week later, you see the process of implantation in the uterus and then further development of the embryo from there. At about week 5, this is approximately what the developing embryo would look like. So here is the head. Here are some pharyngeal structures, which become all sorts of different things around the head and the face. This is what will become the heart. You can see a little limb budge just barely starting to form. You can see that there is still a post-anal tail at this point. Here is the umbilical cord, and there is still an external yolk sac. Okay, so that's kind of where we are starting our story. At this point, there are 2 pairs of ducts in genetic males and genetic females. Same duct system in both types of embryo. And these ducts are called the paramesonephric ducts, right? Formerly called Mullerian. I'll kind of go back and forth because you still hear them referred to as both to this day. And so what we're looking at here is kind of the anterior view. Here is the left lateral view. So this guy's kind of been rotated around. So we're looking at the front of him or her. And here we can see the paramesonephric or Mullerian ducts, one over here and one over here. Okay, we can see the mesonephric or Wolffian ducts, right? That is right here, right? We can see both of them are kind of joining together around in this area. This area is called the urogenital sinus. And so essentially all of the genital or the reproductive structures as well as kidney structures are all coming together and kind of mixing their contents into this area right here. We see this is the cloaca, and so at this phase of development, even the digestive system kind of mixes its contents together, right? Same shared space between reproductive, urinary, and digestive system at this time. Okay. To zoom in on just these 2 types of ducts, I'll actually be walking you through this diagram a little bit throughout this video. Here we can see that the Mullerian or paramesonephric ducts are right here. Okay, so a little bit more lateral as you go up into the abdominal cavity, and then the Wolffian or mesonephric ducts, those are here in blue. Okay, again, they all kind of join together here at the urogenital sinus. Okay, what we also see at this point in development are undifferentiated bipotential gonads. All right, so what the heck does that mean? These are undifferentiated, that is, they have not yet kind of reached that fork in the road where they will develop as male gonads versus female gonads. Okay, so that when I talk about gonads, I'm talking to the testes in males and ovaries in females. And so these are undifferentiated. They are bipotential, so they can become one of two different things, testes or ovaries. And so if we look a little bit closer in here, so this is, it's called the gonadal ridge. This is that undifferentiated bipotential gonad. And so if we look in this diagram down here, again, we'll be looking at this in a little bit more detail. This is what will become either the ovary or the testis, depending on the different hormones and signals that are received during the next week of embryonic development in this case. And so there is the gonad. Within the gonads, we have a couple different types of tissue here. First of all, we have germ cells. And when I refer to germ cells, I'm referring to these cells that can grow up to become either sperm or egg. Okay, so they have all of the genetic material of the entire organism, so this entire developing embryo. And when triggered to, they are going to undergo the process of meiosis, right? Ultimately resulting in a haploid or one copy of DNA cell that will be either a sperm or an egg. Okay, so we have all of these germ cells that have actually migrated into this area. We see them here as little red dots. And then surrounding that is a bunch of extra tissue. It's derived from mesenchyme tissue, so that connective tissue from which all other connective tissues are derived. And so we just have a couple different populations of tissue that is surrounding the germ cells. Okay, so that's week 5. At this point, males and females are exactly the same. And so that story changes in about week 6. Okay, in week 6, there is a switch that is turned on in the case of individuals with Y chromosomes only. Okay, so what we can see here are the two genotypes. We have an X and a Y in males, and an X and another X in females. Okay, so on the Y chromosome only, there's a little gene way up here in the short arm of this chromosome, which is the switch to turn on the male phenotype or the male appearance, the male structures. Okay, this gene is called the sex-determining region of the Y chromosome, right? So very obvious, very straightforward name here. And this is going to be expressed. It's going to be turned on, switched on, not in the heart, not in those developing limb buds, but in the gonadal cells. Right? So in all of that tissue that's kind of surrounding the germ cells in the bipotential gonad. Okay? And so when this gene is expressed, that is, it is transcribed in the nucleus, translated out in the cytoplasm on the ribosomes, the resulting protein is called testis determining factor, or TDF. And so what this protein does is it actually is going to signal the surrounding tissue cells, right? So signal the cells of the gonad to become what are called Sertoli cells, or they've historically been called Sertoli cells. But you know anatomy, right? Why name something once if you can't name it 2 or 3 times? And so they're also called cystenocytes. So they are literally cells that sustain developing sperm. Or sustenaculars, so literally like the sustaining cells, or also called nurse cells. Okay, so these cells, again, surrounding the germ cells, so the soon-to-be sperm cells, are going to turn on different genes and ultimately, right, they will be identified as Sertoli cells. And so once these cells are kind of turned on, they're activated and differentiated, they are going to induce some other gonadal tissue to become the Leydig cells. And again, multiple names here, but the Leydig cells are also called the interstitial endocrine cells. And so as we're going to see on the next slide, the Sertoli cells are kind of the Sertoli cells surround the germ cells, the sperm, and then the Leydig cells are going to be around them. Okay, so let's see what I'm talking about here. Okay, so here we have that same image that we saw a few slides ago. This is the paramesonephric duct or the Mullerian duct, and then this over here is the mesonephric duct or the Wolffian. This is the undifferentiated structure, right? So week 5, males and females look exactly the same. Okay, here is that gonadal tissue here. And so what the testis determining factor protein does is it is essentially going to trigger these structures right here to grow further into what we can now call the testis, right? They're going to kind of surround these germ cells, okay? Ultimately, what they're going to develop over the next couple weeks to next couple months and further into development even after birth is a series of tubules, okay, that are holding the sperm, okay, or the developing sperm, that is. So here, if we take a section through these little tubules, again, which developed from these structures here, growing into the gonad, into the testis, surrounding these germ cells here. And so this is the tubule. If we take a section through the tubule, we see that these are the germ cells, right? We're going to call them spermatogonia from now on. So the spermatogonia are now surrounded by these other cells right here. So this would be the nucleus, and the cell is just kind of like squished in between the developing spermatogonia. Okay. These cells that are surrounding the spermatogonia are called cystenocytes, right? So they are what are literally activated by testis determining factor. Okay. What they're doing, and we'll learn more about this later, what they're doing is they are supporting those germ cells, those spermatogonia, and then, during the process of mitosis, they are supporting them, they are nourishing them, they're essentially nursing them. Okay. Thus the name nurse cells. What they're also doing, and in fact, even more important to our story now, is they're releasing another type of hormone. This hormone is called the anti-Müllerian hormone, or Müllerian inhibiting hormone, or Müllerian inhibiting factor, right? So lots of different names, all meeting against the Müllerian ducts. And so what we're going to see is that this hormone released by the cystenocytes is going to induce the breakdown of this more lateral duct system right here, again, only in males. And this is the paramesonephric duct, and it is going to be breaking down. So within a couple weeks, what you see is kind of the, like a ligament left over from the previous Müllerian duct, and the blue duct here, right? This is the mesonephric duct, or the Wolffian. That one is still remaining. Okay, so males still retain the Wolffian duct. Females, as we're going to see, maintain only the Müllerian. Right, so cystenocytes, which differentiate due to testis determining factor, right, directly related to the Y chromosome, they are not only going to form a bulk of the seminiferous tubules, right, that are producing the sperm, holding on to those spermatogonia, but they are also going to release anti-Müllerian hormone. Also, anti-Müllerian hormone is going to induce some other cells from, kind of the surrounding tissue here, already existing within the gonad. Those cells are going to be kind of told to turn into, right, grow up into what are called the interstitial endocrine cells, or also called the Leydig cells. Okay, so those are here. This is the edge of the seminiferous tubule. What we're going to see later is that these spermatogonia undergo meiosis and produce 4 independent sperm, kind of like moving into towards the lumen of the tubule. Once the sperm are produced, they're going to be pushed along these tubules and ultimately can be ejaculated via the vas deferens. Okay, so that's where we're going with this. So the cystenocytes, or nurse cells, are facilitating that process and guiding that process, but also in between the seminiferous tubules, so in all of this extra space here, these interstitial endocrine cells are going to be producing hormones, right? They are endocrine cells after all. Okay. The main hormone that they're going to be producing, which is also relevant to our story right now during about week 7 or so of development, and that is they are producing testosterone. Okay, so move myself over here. Under the influence of testosterone that is produced by the Leydig cells, or interstitial endocrine cells, The Wolffian or mesonephric duct is going to be retained, right? Remember, the Müllerian duct disappears under anti-Müllerian hormone influence. The Wolffian duct is going to stay, and it is going to further develop. In fact, it is going to develop into the male duct system. And so what I have here is this is the undifferentiated, so about week 5 embryo. Here is the differentiated, so this is a male, right? Genetic male, and now going to be a phenotypic male. And this is an adult male. And so what we're going to do is just walk through the different structures of the Wolffian duct on both the fetus and the adult male. Okay, so first, kind of right up here, the most or the closest part of the Wolffian duct to the testis is going to develop into the epididymis. Remember that in adult male, the epididymis is on the posterior side of the gonad of the testis, and this is what kind of stores sperm and helps them to mature until they are ejaculated. Okay, this tube right here is the vas deferens, again, another structure that develops from the Wolffian duct. Okay. Remember that the vas deferens passes up and over the pelvis behind the urinary bladder. All right, so that's the urinary bladder right here. And posterior to the urinary bladder actually expands to form the seminal glands. Down here on the fetal male, you can see that the Wolffian duct kind of expands a little bit, produces this little, what is called an appendage. All right, so this outpocketing off of the tube itself, and that is called the seminal gland or the seminal vesicle. Finally, not really terribly obvious over here, but on the posterior side of the adult male bladder, you can see that the two seminal glands join together, right? Or the two seminal glands kind of joining together with the vas deferens, produce this little structure here called the ejaculatory duct. And so this is where the fluid of the semen is going to be held immediately prior to ejaculation. Okay, so again, all of these structures, the male duct system, is all derived from the Wolffian duct after the Müllerian duct starts to break down. Okay. Something else that's happening here that you might be wondering about. So essentially, the testes and the ovaries, right? So that undifferentiated bipotential gonad, all of those structures begin way up in the abdominal cavity. And so ultimately, the gonads are going to migrate to different places for males and females. In the case of males, the testis is actually pulled down, right? We can actually see it a little better here. The testis is pulled down into these little outpocketings of skin right here. This is called a labioscrotal swelling, and we'll look at that on the next slide. But essentially, this green ligament shortens, pulling down the testis and what will be the epididymis, pulling down, down, down into these little skin swellings. And so this ligament structure is called the gubernaculum, derived from the Latin word that means helm or something that guides. And so literally the gubernaculum is guiding the descent of the testes down into the scrotum, right? Which is what these little outpocketings of skin are going to become. Okay, so that leads us to talk about the external genitalia development of the male. Now, in order to develop structures into the male pattern, right, kind of like the same structures for male and female, but to develop them into a male pattern, it's not testosterone that drives this process, but actually a derivative of testosterone. Okay, so there is an enzyme called 5-alpha reductase that is going to convert testosterone into what is called DHT or dihydrotestosterone. Okay, so just a fairly simple conversion process, but you need this enzyme in order to make this transition possible. Okay, and so if 5-alpha reductase is expressed, testosterone is converted into DHT, and DHT is going to bind to DHT receptors on structures within the genital region. All right, so within the inguinal region. Okay, same structures for both male and female during this sexually indifferent stage, but under the direction of DHT, these same structures, right, for male and female, we'll go over the female part here in a couple minutes, but these same structures are going to grow at different rates, and therefore produce different patterns, different anatomy in males versus females. And so each one of these structures is color-coded so that we can kind of follow the progression of this particular tissue all the way through, fetal development. Okay, so here is the genital tubercle. So tubercle just means like little bump. And so on this little bump, again, same males and females, under the influence of DHT, this little bump is going to enlarge faster than any of the surrounding structures and develop into the glans penis, or the head of the penis, as well as the main body of the penis as well. Okay. Ultimately, this tissue is going to fold around what is called the urethral fold. Okay, and so here, the urethral folds are actually going to be promoted to grow fairly quickly as well. These are going to become the corpus spongiosum. Okay, so kind of wrapping around the urethra. These are not erectile bodies, and so they essentially leave the urethra open, the spongy urethra open, during an erection to facilitate the process of ejaculation. The erectile bodies, right here, are derived from the genital tubercle, and they are what become engorged with blood during an erection. Okay, these folds right here in yellow, these are called the labioscrotal swellings, and we actually saw them on the previous slide. The gubernaculum is attached to these labioscrotal swellings in both males and females. And so under DHT's direction, if there is DHT, because there's testosterone, because there's SRY, these swellings are going to develop into the scrotal sac. Okay, so the skin will develop at a rate appropriate for the descent of the testes. Okay, so again, undifferentiated structures under the influence of DHT are going to grow at different rates, fold in slightly different ways to ultimately produce the male phenotype. Now, I want to pose a question for you. What do you think would happen if there was deficiency in 5-alpha reductase? Okay, so you are an XY individual, so genetically male, but your gene for the 5-alpha reductase enzyme is mutated, right, in such a way that there is no DHT. What do you think would happen? Okay. Ultimately, individuals with this condition, and this is not a hypothetical, individuals with this condition are born with female genitalia. Okay, so later on in this video, I will show you how these structures develop into the characteristic female shapes. But ultimately, without DHT binding to receptors on these same exact structures, the default is female structures. Okay, and that's internal and external. Okay, so female genitalia at birth. Okay, in the testes, however, there are Leydig cells. Okay, so the SRY gene is still present. It is an XY individual, and so the gonads will still develop into testes. There will be Sertoli cells. There will be Leydig cells. They will develop into spermatogonia. Okay, so individuals with a deficiency or mutation in 5-alpha reductase have female external genitalia, but male gonads. Okay. And so this condition is called testes at 12, okay, or guevedosis, because this was found in an isolated population in a place called Guevedosis. And so really the understanding of this condition was based on individuals in this area. And so what is probably most startling, right, why they called experts in was because, again, these individuals have female genitalia at birth, but then during puberty, there is an increase in testosterone because, again, the testes are male, right? The gonads are male, and so the testes are what produce testosterone, and so the increased testosterone is going to lead to, male characteristics after puberty. And so what happens in these individuals is that, they grow up until they're whatever 10, 12, 14 years old, and then all of a sudden their testes drop, right? Rather startling, I would argue, and I'm sure they would agree. And so from puberty on, these individuals are phenotypically male. So there's an enlarged penis, which was formerly the clitoris. The testes drop into what used to be the labia majora. The voice deepens, muscles are larger, facial hair develops. So everything that you would associate with being male develops only after puberty. Okay. And again, this is just due to a mutation in 5-alpha reductase. So what if there is no Y chromosome? Okay, so we've talked about this genotype here, 1X, 1Y chromosome. What happens if there is no Y chromosome? There's only 2X chromosomes. Right, so we should be able to note that without this Y chromosome, there is no SRY gene. Without the SRY gene, there is no testis determining factor. And without testis determining factor, the testes are not going to develop. Right? Without the testes, there are no Sertoli cells. Right? No Sertoli cells means no anti-Müllerian hormone. As we know, anti-Müllerian hormone promotes the degeneration of the Müllerian duct, and so without this, there is no degeneration of the Müllerian duct. Right? Also, there is no stimulus for Leydig cells to differentiate. Without Leydig cells, there's no testosterone. No testosterone means no male duct system, so no epididymis, vas deferens, seminal glands, or ejaculatory duct. Also, without testosterone, there's no dihydrotestosterone. Okay? Without DHT, there's no external male genitalia. Okay, so kind of the default system here, right? In the absence of this Y, what will automatically proceed is the development of what you would associate with female characteristics. Okay, so an XX individual does not have the SRY gene, and so there's no testis determining factor. Without TDF, ovaries are automatically going to develop. With ovaries, ovaries do not produce testosterone. Right? In the absence of testosterone, the Wolffian duct is automatically going to degenerate. Okay, so the only thing that maintains that duct system, that Wolffian duct system, is the presence of testosterone. Without testosterone, Wolffian duct disappears, and the Müllerian duct is going to be retained. Right? Again, this is the kind of default here. Right? No anti-Müllerian hormone means Müllerian ducts are going to remain. Right? They are going to become the duct system of the female. Okay. So, in the absence of SRY, right, in terms of anti-Müllerian hormone and testosterone, the tissue in the gonad is going to develop into not nurse cells, not Leydig cells, but into follicular cells. Okay, so once again, I want to point out this here. This is the undifferentiated gonad. Here are the germ cells. Here's that surrounding tissue. Remember that these little tubules developed into the cystenocytes, right, the seminiferous tubules. This tissue over here developed into those Leydig cells, which produce testosterone. Here, all of that stuff isn't happening. Instead, what is going to happen is that this duct system, right, we know that that is the mesonephric duct or the Wolffian duct. This is going to automatically break down, right? So this here, and the tissue that is surrounding the germ cells here, right? This tissue is going to develop into what are called follicular cells, right? So differentiating into follicular cells, right? So if we were to zoom in, on one of these germ cells here, right, after differentiation, right, the normal old default system here. These little red guys, again, are the germ cells. In females, we call them oogonia, so over the egg, right? Like big egg here. And the tissue surrounding them are follicular cells. Okay, so if we zoom in on that, what we would see is this. The oogonium is this circle here in gray, right? The oogonium is actually going to start the process of meiosis, right? So it's going to kind of bundle up its chromosomes and get ready for that first meiotic division, that first step in making an egg, right? An actual egg, not just like a stem cell that will become an egg. Okay, and so here we see this is the nucleus, and the chromosomes are all bundled up. They are ready to go. Okay, but in females, not in males, this doesn't happen until a very long time later. After puberty does meiosis start. In females, this oogonium is going to start the process and it's immediately going to stop, right? So it gets ready and then it's going to pause in the very first step in meiosis, which is called prophase 1. Okay, and so the cell we then call a primary oocyte. Okay, and it is actually going to be arrested in prophase 1 for the next at least 10, 12, 14 years, but maybe until the woman reaches menopause. Okay, so essentially, these cells are arrested in meiosis until they are selected for ovulation and then potential fertilization. Okay, so they might be stuck in this step for a very, very long time. Okay, surrounding what we now call the primary oocyte is a single layer of cells, again, derived from the tissue that is already in the gonad. These cells are called follicular cells, and they form a shell, a single layer thick, around each and every primary oocyte. Okay, we call the shell itself, so this group of follicular cells, we call it a primordial follicle. Okay, and we're going to see how that changes in preparation for ovulation later on in the woman's life. Okay, so ultimately, the whole ovary, what it consists of, right? So here's an adult ovary. There's a lot of extra tissue here, right? So just like connective tissue holding onto blood vessels, lymphatic vessels, and really giving structure to the gonad. And then within that, so suspended within that, at this point in development, there are groups of these primordial follicles in what are called egg nests. So they're actually like grouped all together in egg nests. Later on, and we're gonna get this in another lesson, these primordial follicles are going to be selected to keep on developing. Right, so we see the follicle gets bigger and bigger and bigger. The oocyte within is going to complete the process of at least meiosis 1. And ovulation is actually those cells, right, that follicle bursting and more or less spitting out this oocyte towards the uterine tubes or the fallopian tubes. And so let me cut back to this over here. Here's the ovary, okay, this gonad. Mesonephric duct, the one that was actually closer, disappears. The Müllerian duct is actually farther away, but it is retained. And so we see that even at this step, the ovary and the Müllerian ducts are not touching. Right? We can even see these little finger-like projections here. And so ovulation, this process right here, actually involves spitting the oocyte out, right? Ovulating the oocyte out into this space within the pelvic cavity, or ultimately within the pelvic cavity, and hopefully getting that egg over here into what will become the uterine tube. And so that's why those fimbriae are so important, these little finger-like projections. They are literally like, pulling that oocyte. Come on, you can make it into the tube. Okay, so again, in the absence of SRY, right, which is on the Y chromosome only, there's no anti-Müllerian hormone, and so the Müllerian duct is retained, mesonephric duct disappears. Without testosterone, right, we don't have the development of this tissue within the gonad developing into cystinocytes and Leydig cells. Instead, these cells are going to differentiate into follicular cells. Right? And each one of those developing eggs, in this case primary oocytes, is surrounded by follicular cells to form primordial follicles. Okay. Difference between males and females at this point in the game. Note that in the testes, I did not say meiosis was begun. Right? Essentially, those stem cells, the spermatogonia, were just kind of hanging out. They have not started the process of meiosis yet. They have not replicated to produce a lot of them. In females, on the other hand, meiosis starts here during development. Right? So literally, the egg that made you was created, right, was underwent mitosis and then meiosis in your grandmother, right? So in your mom, while your grandma was still pregnant with her. Okay, so you have been around a very long time, or at least part of you has been around a very long time. Okay. When the female is born, she is actually born with all of the oocytes, right? All of the potential eggs she can ever have, right? Males, more sperm are produced every single day from puberty on, usually until, for the rest of his life. But in females are born with a finite supply of these primary oocytes. Okay, so let me kind of zoom back to this diagram. Previously, we looked at the undifferentiated gonad. Okay, under SRY expression, the gonad differentiated into a testis. So there were Leydig cells, there were Sertoli cells. Okay, and because of the secretions of both of those, the Wolffian duct was retained and the Müllerian duct broke down. Okay? The Wolffian duct became the epididymis and the vas deferens. Okay? And the gubernaculum pulled the gonad and the epididymis and the resulting vas deferens down into these labioscrotal swellings, right? Which in a male become the scrotum, as we know. Okay. Without SRY expression, the Müllerian ducts are going to develop into the female duct system. Okay, so some of these ducts you might not think of as ducts, but they really did start out as little tubes. Okay, so here the Müllerian duct is retained, Wolffian duct breaks down. The Müllerian duct way up here becomes the uterine tube or the fallopian tube. Okay, note that way down here, the Müllerian ducts come together, and what's going to happen is the two ducts kind of zipper together, and the wall of the tube between them is going to break down, right? So apoptosis of the cells in between. And so what happens then is that essentially these two tubes open up into a larger, somewhat cavernous space, which is the uterus, right? So again, the uterine tube, here are the little fimbriae. Uterine tubes join together, zipper up, and form this larger uterus. Okay. And so note that, at this point, the Wolffian or mesonephric ducts are still kind of present. Sometimes there are still remnants of the Wolffian duct in females and of the Müllerian duct in males. Okay, so there are actually little structures here that are present in some people. Okay, the gubernaculum in females does not pull the ovaries all the way down into the labioscrotal swellings. Instead, it kind of degenerates and forms into like a little ligament, which is attached over here and still into those labia, into adulthood. Okay, so let's take a look at this in another way. In the beginning of this video, I showed you guys this is the anterior perspective of the developing embryo and then soon to be fetus. Right, the two pairs of ducts come together at the urogenital sinus under SRY's influence, so anti-Müllerian hormone and testosterone. The 5 to 6 week old undifferentiated embryo is going to start expressing these things, these different hormones, which is going to drive the development in the male direction, right? And ultimately, this is at birth. Okay, without SRY, right, this is the default condition, right? As long as neither of these are being detected or they're being detected at a really low level, this development is going to occur. So let's just walk through the different homologous structures. So same structures, different orientation. Okay, so here's the gonads, the testes, and the ovaries. Okay, note that the paramesonephric duct degenerates in males. Mesonephric duct degenerates in females, and so what's left is the tube system of the males, epididymis, so nice and squiggly, lots and lots of space to store sperm. The vas deferens, little appendage popping out here, the seminal gland, and joining together to form the ejaculatory duct. In females, the paramesic duct forms the uterine tubes. Okay, uterine tubes fuse together to form the uterus and also the upper third of the vaginal canal or the vagina. Okay, and again, we can see the same structures into adulthood. The testis and all of the associated tubes are pulled down, down, down until they are outside of the pelvic cavity. Essentially, the idea here is that sperm like it best where it's a little bit colder, so colder than normal body temperature, and also other things like alkaline and some other conditions. And so keeping the testes outside of the body actually increases fertility. Okay. In females, right, here's the fallopian or the uterine tube, uterus where the two join together. And here the urinary bladder we can see has been moved out of the way. Okay, so this is in the pelvic cavity, this is out of the pelvic cavity, and in the pelvic cavity. Okay, what about the external genitalia? We have already examined how these structures under the influence of DHT grow to produce a male pattern of external genitalia. Without DHT, right, or if DHT cannot be detected, right, if there is a mutation in the receptor for DHT or something, external genitalia of the female is going to develop. So again, the same exact structures develop in a slightly different shape, and usually smaller, but same structure, same color coding. Okay, in the male, right, the blue structure here enlarges to become the glans penis and the body of the penis. In the female, becomes the glans clitoris, so essentially the head of the clitoris. Okay, still an erectile body, right? So remember that the glans penis and the body of the penis, the corpora cavernosa, that is the erectile tissue of the penis. Same exact thing in a female. The clitoris is an erectile body, largely the same in structure as the corpora cavernosa. The urethral fold, remember this forms the corpus spongiosum in the male to assure that the spongy urethra remains open during an ejaculation. In a female, the urogenital sinus is going to somewhat fuse together and leaving a tiny opening for, so here you go, for the urethra, for the vaginal orifice here, and surrounded by two lips of skin called the labia minora. Okay. Finally, the labioscrotal swelling literally tells you exactly what it's going to become. Labia and scrotum, same structure, DHT or not determines what direction it's going to go. Okay, so the labioscrotal swellings become the scrotum in males, right? So eventually the testes are going to be pulled by that gubernaculum down into these flaps of skin. So covered with pubic hair in males, same exact thing in females. The labioscrotal swellings develop into the outer lips. Again, these are the hair-covered ones, same exact pattern as in the males, and these are called the labia majora or majus. Okay. And so one other note that I'd like to make about this process, everything that I've been talking about so far has been occurring between approximately week 6 or so until maybe around week 12. And so all of the patterns, all of the structures are essentially laid down during that period. And so this window is called a critical period. It is a window in the development where the development of a specific system is going to begin, and establishes the pattern for that system, right? Whether we're talking about the brain or we're talking about the heart or we're talking about the ears, whatever we're talking about, there is a specific window during fetal development that that organ starts to develop, and like whatever structure it grows into then is what essentially what you are working with for the rest of your life. And so it has these enduring structures or these enduring properties. And so it is during this period, what is called a critical period, that any variations in your genes, right? So different alleles that you have, different variations, different gene expressions, right? So whether we produce 5-alpha reductase a lot or a little, that is going to heavily influence the structures of your genitalia, your external genitalia, for the rest of your life. Okay. Also, during this time, the levels of different hormones, right? Keep in mind that during fetal development, mom is producing primarily female hormones like estrogen and progesterone, right? So the hormones that would be dominant in a female as opposed to a male. And so if there are any so what I'm getting at here is that it is kind of the fetus's job to produce their own testosterone if they are genetically male. And if there are any variations in hormones, so any exogenous hormones, any chemicals that act like hormones or block the activity of hormones that mom is exposed to, that is going to have the biggest effect if those exposures or that variation is experienced within the critical periods. Okay, you'll see where I'm going with this here in a moment. And so after this critical period closes, you can no longer really change the trajectory of your development, right? So towards male or towards female without surgical intervention. So no means of naturally going in one direction or the other after the groundwork, after the foundation has already been laid down during this critical period. Okay, and so exposures later, particularly during sensitive times, for example, during puberty, when you have a lot of bodily changes, and so hormones once again become critically important to be at just the right level to make sure that you develop, breasts or facial hair, right? Not both, or to the extent that was intended. Okay, so again, there are critical periods that lay the groundwork, that establish structures for the rest of your life. Any exposure or any variation in gene expression or hormones after that period are just acting on existing structures. Okay. And so there are periods of more sensitivity. So actually, what the image we see here is showing us is the critical period, for example, of the genitalia. This is that most critical period. And then, this is when SRY is expressed or it's not, right? So really laying the groundwork for male direction or female direction. But then, there's still a long period where you're really sensitive, right? So again, any variations in hormone or gene expression, even during this yellow period here, during the sensitive period, can also have reasonably significant effects, but not nearly as much as variations during the critical period here. And again, puberty is another one of those sensitive times. I also want to point out the central nervous system. So levels of different sex steroids, for example, testosterone, dihydrotestosterone, and estrogen, are really important in, reaching that fork in the road and going either in the male direction or the female direction, right? So during this critical period here. But the central nervous system has a much longer critical period. Okay, so here we can see very long critical period all the way, you know, into, later into the pregnancy and remain sensitive throughout the rest of pregnancy and the beyond. And so what I'm getting at here is that, first of all organ systems have different critical periods. Some are long, some are short, okay, and some are spaced out differently throughout the pregnancy. The nervous system in general takes the longest time to develop, and it is also heavily influenced by steroid hormones, including testosterone and estrogen. So these hormones can drive the development of the brain into, what we would stereotype as a male or a female brain. In reality, male and female brains are just a myth, right? It's all a spectrum, but generally, hormones at this time can push a brain into having more what we would call female characteristics, or push more in the direction of what we would call male characteristics. Again, it's all a spectrum. There is no such thing as a male and a female brain. Okay. Furthermore, it's not just steroids like testosterone and estrogen, but cortisol, stress hormones, are also a potent force in the development of the brain, and they can interact with sex steroids, right, testosterone and estrogen, in such a way that it is going to change masculine and feminine behaviors for the rest of your life, right? So mom stress levels, baby stress levels heavily influence how the brain is developing throughout pregnancy. Okay, so I could talk lots and lots about that. Absolutely fascinating topic, how the brain develops in that context. But what is relevant to this this video here is the critical periods and the levels of androgens, right? So testosterone, DHT, etc., and estrogen levels, and even the sensitivity to these things. So how well do the receptors for testosterone versus estrogen actually bind to these hormones? How these variations genetically and in terms of gene expression, how do they influence sexual differentiation? Of course, I keep mentioning that there is kind of a fork in the road, right? Is SRY expressed or is it not? But it's not all or nothing, right? There is considerable variation in how much of these hormones are produced, the shape of the receptors, right? Maybe even mutations in the genes that code for aromatase or code for 5-alpha reductase, as we already saw, right? So any kind of genetic or even epigenetic variations can significantly affect sexual differentiation, leading to not just male versus female, but a spectrum. For example, very little fetal androgens, right? Very little testosterone or DHT, as would be the case in a genetic female. The resulting external genitalia, right, and actually development of the entire perineum, is approximately like this. So this is the structure of the phallus on the anterior side of the perineum, right? So this would be the clitoris. There would be no urethra, no urethral opening in the clitoris, right? Moving posteriorly, vaginal orifice. Okay, moving posteriorly to the vaginal orifice would be the anus. With the most fetal androgen, okay, so this is an SRY condition, plenty of testosterone, plenty of DHT, plenty of all the enzymes that make them, all the way anterior, the phallus. Okay, so this would be the penis in this case. The urethra opens at the tip of the penis, so at the very end of the glans. The perineum, there is no orifice, right? And the anus is considerably far from the phallus, okay? However, it's not all or nothing, as I've been describing. We kind of talk in terms of extremes, but in reality, because of the genetic and hormonal variation that I've discussed, there is a spectrum of these phenotypes. Okay, so as androgen levels increase, right, or sensitivity to androgen levels. Okay? What you see is slightly different structures. Okay, so with a little bit of androgen, what you see is maybe the tip of the or maybe the urethral orifice actually may exit in the phallus, so in the clitoral tissue instead. Right, the vaginal orifice is much closer to the phallus than it otherwise would be in a case where there would be no or very, very low androgens. Okay, and as you continue here, what you see is the distance is going to change between the anus and the phallus. And something else that I want to point out here, in these intermediate phenotypes, there are conditions where the urethra, because of the way that these urethral folds fold under DHT versus no DHT or a little bit of DHT, because of this urethral folding, sometimes the urethral orifice, so where the urine, where the semen would eject from the body, sometimes that is not all the way at the tip of the glands. Right, so let me talk about a couple conditions here. Androgen insensitivity syndrome. Okay, in this syndrome, an XY individual, so has SRY, therefore, should be producing testosterone and DHT. In this case, XY individuals present more as a female, right? And we're talking about anatomic female. We are not talking about gender expression. Okay, and so what is occurring within the symptom or within this syndrome is that the androgen receptor has, is not shaped in such a way where it is super sensitive to androgens, that is testosterone and DHT. Okay, so in some cases, the androgen receptor may not bind to androgens at all. Okay. And so androgen insensitivity syndrome, AIS, can be classified as mild if it just like binds, mostly, but is a little bit sluggish, right? So maybe like this condition here, right? Complete or partial. So a lot of different levels here. And of course, that's going to determine kind of where on this spectrum the individual is. Okay. Exposure to different chemicals during a critical period, right? So we're talking about mom is exposed to a chemical. That chemical crosses the placental barrier and therefore influences the development of the embryo and the fetus during its critical window, right? So during the baby's sexual differentiation process. Many chemicals that we interact with on a daily basis, things like BPA, which yes, is out of our water bottles, but it's still in receipt paper. It's still in the lining of canned goods. Atrazine and other types of pesticides, right, are on your food unless you're eating organic food. Various types of organic solvents, in all sorts of different products, including a lot of hand soaps. Okay, so lots of different chemicals that generally make our lives easier, but at the same time, they can mimic hormones. So essentially, they can look like testosterone or estrogen, even if you yourself are not making those hormones. They can block testosterone or estrogen, right, from binding. So maybe they would bind to this androgen receptor and block androgen from actually binding and, driving the development in this direction as opposed to staying close over here. And so lots and lots of different examples, but in general, what these do is they are mimicking or blocking the endogenous activity of your own hormones. And so, they can drive this development in this direction or in this direction, regardless of your genetic sex. And one condition that is seen with increasing prevalency over the last couple decades is what is called hypospadias. So what this is essentially exposure to things like BPA and atrazine, right? Again, a pesticide that is used agriculturally and residue is still on the food that you eat. What exposure to these things do is they, drive the development, the spectrum in this direction. And so hypospadias is when the urethra exits not at the tip of the phallus, not at the tip of the penis, but instead on the inferior side. And so this can have, kind of like inconvenient side effects for urination, but also this can lead to fertility problems because, an ejaculation is not, propelling semen into the uterus, right? It is propelling it into the side of the vaginal wall. And so this leads to fertility problems in addition to, issues with spermatogenesis in general, which the same chemicals are also influencing. Okay, another example here, congenital adrenal hyperplasia. And so the congenital here means you're born with it, so some type of genetic condition. Adrenal, so this is a problem with your adrenal glands. And the hyper here is telling us that essentially your adrenal glands are overactive. And so the adrenal glands, we know, produce aldosterone. We know they produce cortisol, the stress hormone. But they are also able to produce androgens, so masculinizing hormones. Okay, so both males and females. And so if your adrenal gland is overactive, right, it is producing too many androgens during that critical window of your development. And so what that can lead to is, essentially, if we go back here, it can lead to shifting a genetic female closer in this direction. Okay, what can also happen is that, here we'll go back to this. It's called persistent urogenital sinus. So remember, early on in development, the urinary stuff, the reproductive stuff, and even the digestive stuff is all going to like mix together in this one space called the cloaca. Over development, or over the process of development, the digestive stuff kind of separates first, and then in females, right, not in males, right? Remember that males kind of share urogenital stuff, right? The urethra accepts fluids from both the urinary and the reproductive structures. But in females, those two organ systems are completely separated, right? Or they are under estrogen dominant control. All right, so this is normally what happens. The urethra is completely separated from the vagina, which is completely separated from the rectum. In the case of congenital adrenal hyperplasia or a series of other developmental anomalies due to differences in hormone levels, okay, the vaginal canal does not separate from the urethra. Okay, and so this this would also lead to infertility because intercourse is not, not possible, or at least not comfortable in this, or given this anatomy, right? There are surgeries that can separate these two structures out, right? But again, this is caused, right, by essentially genetic female being pushed in this direction by too many androgens, right? Too much testosterone. Okay. Congenital adrenal hyperplasia also increases androgens, so testosterone, in genetic females to genetic male levels. And so if the levels are high enough during that critical period, essentially, even though there isn't a testis producing testosterone, the adrenal gland is producing testosterone or other androgenic hormones. And those hormones, right, can be converted into DHT, and DHT can therefore drive the development of external genitalia in the male direction, right? So growing into that male pattern. All right, so that is all I have for you guys today. Thank you guys so much for watching. I hope that you learned something really interesting that you didn't know before. Thank you.